Good morning and welcome to our first joint webinar co-organized between NanoMed Spain and NanoMedicine Austria. Today's webinar is focused on the topic fostering international collaboration in nanomedicine, spotlights from the Spanish and Austrian nanomedicine platforms. So the idea was really to get to know the Austrian and the Spanish nanomedicine communities and create collaboration between the specific research topics and innovation topics. And it's a great pleasure today to host this webinar, and big thanks also to NanoMed Spain for the great collaboration in setting up everything and setting up the agenda. So we are looking forward to great presentations. I hope everybody is also keen on what we have on today's agenda. So you can see we will start with the NanoMedicine Austria platform. So you will have a quick overview of the platform, and then we have scientific presentations from members of NanoMedicine Austria. Then the second part of the webinar is focused from NanoMed Spain. Again, a quick overview of the platform, followed by several scientific presentations from NanoMed Spain members. And then the last part is dedicated to discussions and Q&A. So please feel free also if you have any questions to put them in the chat during the presentations, and we will try to address them properly at the end of the webinar. So it's a great pleasure to also have Josep Samitier joining the webinar, and happily handing over for your welcome words.
Hello and good morning, everybody. Sorry for my short delay. It's a great pleasure for me to welcome all of you to this joint nanomedicine workshop between the two platforms, the Austrian platform and the Spanish platform. For us, it's very, very nice this opportunity to strengthen the links and the collaborations between the two countries. Very active in the field of nanomedicine during the last years, and well, basically this welcome is also a preliminary activity before to receive many of you in some weeks in the European Nanomedicine Platform conference that is located organized here in Barcelona. So I hope that will be the opportunity to join in person many of you in this event to celebrate the 20 years of European Nanomedicine Platform and 20 years also of the Spanish Nanomedicine Platform. So thank you all of you to participate in this workshop. Thank you all the speakers, and please continue with the program and enjoy of this day. Thank you.
Great. Thank you so much. And best wishes also from Graz to Barcelona. So then I will continue with the presentation of the NanoMedicine Austria platform. As said, my name is Susanne Resch. I work for BNN in Austria, and I'm a coordinator of the Austrian Nanomedicine platform. So let me change slides. How did we start? So nanomedicine is definitely a key topic within the BioNanoNet Association, and BNN is coordinating the platform. We established NanoMedicine Austria in 2016, so it's a little bit younger compared to the NanoMed Spain, but also already several years running. And NanoMedicine Austria is the Austrian technology platform addressing the scientific and technological challenges in the nanomedicine field. So we really try to bring together all nano and biomedical experts to promote science, research, and also the translation to the clinics in the end and the market of nanomedicine. So the idea is really to build the community, gather expertise, increase the visibility of the existing expertise in Austria, and also join forces when it comes to funding programs addressing different topics in nanomedicine. As a platform, we do have a vision and a mission, which is stated here on this slide. So the platform's vision is to increase the visibility of nanomedicine as a benchmark for research and innovation at national but also international level, and of course promote the Austrian excellent basic and applied research to strengthen in the end the transfer of nanomedical applications from lab to clinical practice. And the mission is to build references on Austria's nanomedical research and innovation capability and to achieve joint impact through community-driven activities. So more or less what I also already said in the last slide, to create greater impact as a whole community rather than single research organizations. What are the topics in Nanomedicine Austria? So you see in the middle the core topics related to nanomedicine, ranging from diagnostics to medical imaging, but also nanotherapeutics, vaccines, and also regenerative medicine. And what we added are also some cross-cutting topics that we believe are also important in the nanomedical fields. So bioinformatics in nanomedicine, but also the nanospecific characterization is, of course, very important. Then also we have expertise in Austria on the targeted and controlled manufacturing of nanoproducts, and also standardization and regulatory topics are important to bring also research to the market in the end. How is the platform organized? So we have a core group that consists of the general coordinator and the scientific chairs of the platform. Then we have the board of members, where all member organizations and experts are gathered. But also we try to reach out to other communities and platforms. So that's also one reason why we organize this webinar today. We believe that it's not only important to be strongly connected within Austria, but also with other countries, with other platforms, also the ETPN, NANOMED SPAIN, etc., and other stakeholders to really increase the impact and also make nanomedicine an important topic on European and global level. The scientific chairs of the platform are listed on this slide. So we have currently 4 scientific chairs: Daniel Baumgarten from the University of Innsbruck, Ramona Jaitler from the University of Graz, Thomas Mohr from ScienceConsult, and Sebastian Schwaminger from the Medical University of Graz. All of them bring in different key topics and expertise, also maybe familiar to the slide I showed before. So Daniel is strong and has expertise in standardization. Ramona has a great experience in controlled manufacturing of nanoproducts. Thomas Mohr is very active in the bioinformatics field. And Sebastian is active in the field of nanomaterials for drug delivery. So it's a nice complementary expertise also among the chairs. Regarding the board of members, we currently have 22 active member organizations. You see the logos here listed. It's a mix between academia, RTOs, but also industry. And generally, Nanomedicine Austria is an open platform. So more or less, anybody who is interested, any expert that has experience in the field of nanomedicine can contribute to Nanomedicine Austria. And we always also try to increase the organizations and get more people on board that are active in the field. We also produced a strategy paper from the platform. So if you're interested to get more details and more information, I also invite you to check it out. It's published on Zenodo. And it also gives the strategy of the platform, what we try to achieve, what are the activities, etc. Today I also want to quickly present the goals of the platform in more detail. So again, as already said, a main goal is, of course, to tackle medical challenges, to achieve a more efficient and affordable healthcare, as well as the development of new solutions to improve medical treatments. But also, of course, as a platform to connect the different multidisciplinary experts in the nanomedical field. and also connecting the different stakeholder types that are relevant along the whole value chain of nanomedicine. Then, of course, create synergies through the cooperation among the different actors. This could be done either through projects or other collaboration types. Then also support the Austrian nanomedical community and link it to the European and global nanomedicine-related initiatives. Enhance also the access of national and international research projects and funding. So this is also definitely a key topic because, as you all know, research usually also depends from public funding. So this is definitely a hot topic. Then also to facilitate the collaboration with industry. So to move out of the pure scientific research at universities, but also close the gap to the industries. And also, last but not least, but also super important, provide support in topics like nanosafety, quality, and also sustainability aspects to meet the regulatory requirements, which are very important. What are the activities then to achieve these goals? So we do have regular meetings and networking activities. We have regular chairs meetings, but also twice a year a board meeting, and we also organize webinars and networking events. Then, of course, we try to refine and update the platform strategy. So the strategy paper I showed before will definitely also be updated at some point. Then we also contribute to relevant position papers, generate and collect possible topics and ideas, and also contribute them to national and international funding agencies. Then, of course, we provide support in the organization of workshops and trainings for students, as it's important to also pass knowledge to the early career researchers. Then we support with grant applications on different programs, so national but also international. And we create outreach to relevant stakeholders, including public communication. So we do have a NanoMedicine-Austria website, a LinkedIn account, we contribute to newsletters, etc. And how are we doing this also? So as I said in the beginning, NanoMedicine Austria is coordinated by BNN. As BNN, we have different services and areas that we provide support in. So for instance, one service area is called Go Compliant. So here it's about regulatory support for medicinal products, but also medical devices and IVDs, specifically with nanomaterials and advanced materials included. So we do have an expert in the team who is really a regulatory expert and can help you with all these topics. Then we have the Go Sustainable area, where it's about making nanomedicine, but also other innovations, not only safe, but also sustainable for humans and the environment. Then we have the Go Smarter area, which is about training and education. So for instance, you can also request us to provide specific training in regulatory aspects. Then we have the go-to-market area, which is all about innovation support and how to bring your research and your ideas really to the market. And in the Go Beyond, it's all about alliances and clustering. So how to create synergies also with other topics. So as BNN, we coordinate NanoMedicine Austria, but we also have other platforms that we coordinate. For instance, the Sustainable Chemistry Austria and also the Microfluidics Austria initiative. So yes, also there we try to create synergies and connect the people so that we have the greatest impact as possible. We also coordinate the BioNanoNet Association. You see here the member organizations listed. We currently have 72 member organizations. Again, here nanomedicine is a key topic, but also other topics like microfluidics, sensor solutions, everything around nano and advanced materials are key topics. And we try to support our members to build consortia for projects, create events together, more or less join forces in this field. As Josep said already in the beginning, there will be the NanoMed Europe end of May in Barcelona. And if you have any other questions or if you would like to reach out to BNN or Nanomedicine Austria, I'm happy to say that my colleagues Johanna and Daniel will be there on site, so you can meet them there and further engage and get in touch. And also there will be the Phoenix Project second satellite event back-to-back with the NanoMed Europe conference on the 30th of May. So BNN is also part of the Phoenix Project, and we will support also this event there on site. So please, if you are at NanoMed Europe, also connect with my colleagues and potentially join these satellite events, which is called Redefining the Nanopharmaceutical Journey from Bench to Bedside. Great. With this, that was my introduction and presentation about Nanomedicine Austria. I can now hand over to our first scientific presentation from Nanomedicine Austria, which would be Sebastian Schwaminger from the Medical University of Graz.
Hello, thank you, Susie, for this very, very kind introduction. I would very much like to also present some of the nanomaterial science we conduct in Graz, Austria, and would like to start with the first topic. In this case, it's about magnetic molecularly imprinted polymers for drug delivery. So in general, I can repeat a little bit on the general challenges in traditional drug delivery, that we have always an issue that if drugs are administered without targeting, we have systemical actions of drugs on the body and therefore also systemic responses. We might have off-site effects and immunogenicity issues. Therefore, we have a lot of costs and hospitalizations which need to be addressed. Therefore, we actually want to go in a dual way. So we want to, on the one hand side, develop systematically peptides which are able to target tumors. That's one aspect, and the other aspect, we specifically develop controllable delivery agents for the distinct release of different drugs. In the example I will be presenting here, we show the example of cationic peptide for drug treatment. And so our release mechanisms which we are building is based on multiple dimensions. So we always have a magnetic core which can be used on the one hand side for targeting, on the other hand side as a kind of a trigger for distinct release. Therefore, we both have it to distinctly control the size of the particles, being in a size where we don't have big immunogenicity issues, and also can increase the dosage. And about the first thing is why molecularly imprinting and how does molecularly imprinting work? So molecularly imprinting for us is a platform to basically have a distinct affinity adsorbent system, which we can use as a platform for targeted delivery. For the first system, we need a distinct template. And on this template, so the template would be, in our case, basically the receptor of any kind of tumor cell. We have multiple different peptide regions identified, which we can use here as a template. Then around this template, we construct functional monomers. So the functional monomers have this kind of key lock feature. So there will be definitely some interactions. This could be electrostatic interactions or hydrophobic interactions distinctly with the template receptor. And they are always having the possibility to be polymerizable. We then have a pre-polymerization kind of adsorbed physical state complex, and then start the polymerization. So this is a radical polymerization, where we are using radical polymerization starters. have the polymerization around our template, and with a template removal and rebinding, we kind of get our molecularly imprinted polymer. So therefore, we have then a polymer which we can use. In our case, we use glass beads. On these glass beads, we bind distinct peptides which resemble the receptor structures on the surface of tumor cells. With this peptide, we do then a cross-linking initiator and the functional monomers, which are kind of the counterparts to this peptide, and do polymerize them. We furthermore add some trigger material, so PNIPAM, which allows for temperaturely controlling the binding and the release. Therefore, we can actually release our molecularly imprinted polymers by cooling them down. So therefore, the binding will collapse with the temperature change, which can be also used for thermal release within the body, but also can be used for the purification of our systems. That's the basic process we're using. Therefore, we are having a column-based solid phase synthesis. So basically, we can kind of automate our synthesis. So we use glass beads, fill them in a column, put there our receptors, and polymerize them with cross-linkers, and get our molecularly imprinted polymers out by thermal elution. This is one classical peptide receptor for which we've done that is caterin, for the inhibition of cell-cell interactions. So this is a typical epitope on multiple cells, which could be used, which we tested here. Another system which we're using is a drug which was imprinted. In this case, it's the peptide lesiglossin. Lesiglossin is a cationic peptide which leads to the lysis of different cells. On the one hand side, it's about tumor cells for drug delivery. On the other hand side, it can also be used as an antibiotic for the lysis of different bacteria, mainly gram-negative bacteria. So in this case, the membrane disruption distinctly leads to cell death with these kind of peptides, which can be easily imprinted specifically with high affinity, then being rebound kind of to our molecularly imprinted polymer, and then to be used as an ideal candidate for cancer treatment. In our case, we basically used iron oxide nanoparticles as cores, did a polymerization around that, had the kind of polymers. To these polymers, we were then adding this kind of systematic peptide. To the peptide, we did a further polymerization of different groups. In this case, we used polyacrylic acid coated systems and copolymers of polyacrylic acid and metacrylic acid coated particles for the binding and rebinding to our systems. And then we analyzed, figuring out if the polymer really works and is still bound to the iron oxide particles with different techniques. So we characterized the particles with IR spectroscopy as well as with dynamic light scattering, being able to show that these nanomaterials are still in the nanoscale range. In our case, it's usually below 200 nanometers. Usually there was one exception, but usually it's between 100 and 200 nanometers. In our case, we then do a reabsorption of the peptide to the polymer, coated nanoparticles. The easy thing for all our assays, we can use magnetic separation to test them and to verify them. We saw that it's quite easy to get a high load of the peptides to the nanoparticles, and it's not only a high load which is then specifically bound, but it's also not easily released, and therefore being the first kind of part of having an effective drug delivery system with a high load of a drug. And then we further characterized it according to the zeta potential, and checked then the cumulative desorption for our systems, as well then as what happens with washing. Therefore, we saw it's quite a stable formulation for this kind of peptide drug. And we actually saw that with our systems, we were able to even achieve a higher cytotoxicity than just with the peptide alone. While the polymer is completely non-toxic in our system, we showed that the polymer combined with the antimicrobial peptide, we actually have a higher cytotoxicity than just the peptide alone. We explain that by actually having the functionality that way, that it really can kind of peak directly towards the membrane. And therefore, we can conclude that we have a really derived an easy and cheap synthesis for these kind of polymers for drug delivery systems, and are able to enhance antimicrobial activity. We also have seen promising results with cytotoxicity for cancer cells, and would always be happy to collaborate in this field if somebody has similar research questions towards targeted drug delivery where we might be able to help. And with that, I would like to acknowledge my collaborators and thank you all for your patience, and hope to discuss it. Thank you very much.
Great. Thank you very much, Sebastian, for this interesting presentation. And with this, I would already like to hand over to the next speaker, Thomas Mohr, who will talk about bioinformatics meets nanomedicine.
Okay, thank you very much, Susanne, and it's a pleasure to be here. And the thing is, as a short introduction, as I'm Thomas Mohr I'm here as the CEO of ScienceConsult, but I also work at the Medical University of Vienna and the University of Vienna, where I have several teaching assignments in terms of bioinformatics. Yeah, and today I would like to give you not so much a scientific data presentation, but I would probably give you some ideas as to how to apply bioinformatics in nanomedicine and how to combine that. The thing is that in the last years is that the bioinformatics has gained a lot of traction actually in the field of molecular biology. And it offers, as I've listed here, a few key points. So, for instance, the convergence of bioinformatics and nanomedicine has opened new frontiers in precision medicine. Then the bioinformatics offers computational tools to analyze biological data with an hitherto unknown in-depth. And I think so that the combination of nanomedicine and bioinformatics has a very, very high potential. Since I am originally from cancer research, I will now focus on a merger between bioinformatics and nanomedicine in cancer. But I think the general conclusions apply to any pathology. Okay, so how does bioinformatics help in cancer therapy? So first of all, we see several so we can split it in several subfields. So we have, first of all, the cancer diagnosis, the cancer treatment, and the assessment of biological response. And especially as in cancer diagnosis, bioinformatics is especially useful in early detection, patient stratification, and the prognosis of resistance identification. With regard to cancer treatment, we see a huge application in the therapeutic target identification, then the DS design and treatment strategy determination. As especially, for example, in melanoma, bioinformatics has led to a huge improvement in treatment. If you think about, for instance, the BRAF mutation melanomas, where bioinformatics is the discovery of these BRAF mutations via bioinformatics has led to a huge impact on the therapy. Then we have here the TME regulation, the metabolic impact, protein synthesis and differentiation, genomic and pathway alterations, biodistribution, and toxicity assessment of the drugs. And all these fields are covered by several topics of bioinformatics. We have here genomics and transcriptomics, metabolomics, proteomics, glycomics, microbiomics, and epigenomics. And all these so-called omics technologies are connected with a huge amount of data, which have to be analyzed using specialized methods. So to be honest, with a simple t-test, you won't get very far here. And this is actually the field of the bioinformatics stuff. The general concepts in omics data analysis in cancer therapy or any other therapy, therapeutic therapeutic scenario is that we source first the samples. That could be cells, that could be xenograft models, that could be patient tissues, biofluids, so you name it. The next step would be the sample extraction, which would be DNA, RNA, protein, a metabolite, or a lipid, depending on what you're doing. Then we would do some omic sequencing, respectively metabolite determination, which is carried out by next generation sequencing or single-cell RNA-seq, spatial transcriptomic, mass spectrometry, etc. Then comes the basic stuff like differential feature expression analysis, with feature being either a gene, protein, metabolite, or lipid. The enrichment analysis, which puts the identified differentially expressed features into biologic context. Then we have a network analysis and more integrated analysis. And the results of these analyses may lead down now to the clinical application of the whole stuff. For example, patient stratification, as in the BRAF mutation melanoma, the prognosis prediction. Classical example would be the glioblastoma diagnosis, prognosis prediction via the molecular subclasses. Then precision therapy, which also is an example, would be as the BRAF mutation-based precision therapy, and mechanism research. And I will now give you a few examples on how to merge nanomedicine with bioinformatics. The first thing is an example about early detection of cancer. And what is depicted here is a workflow to fish marker proteins out of biofluids. So we see here, so first of all, we get a sample from the patient. And this sample is now mixed with specialized nanoparticles. In this case, it would be a silicium dioxide nanoparticles. And these silicium dioxide nanoparticles more or less catch the proteins out of the sample. And these, to form a so-called protein corona, and these protein sample preparation is then subjected to mass spectrometry. So here, with spectrum library generation, etc. And then we get some mass spectrometry data library. And based on this mass spectrometry data, we then can then see whether this patient may have a cancer, either at health, so either early stage, advanced stage, high risk, or healthy. The next thing would be, for example, a prognosis prediction. In this case, so this is shown on the example of breast cancer and the metabolomics approach. So here we have, again, the sample collection from the patient in form of blood serum. And this is also mixed with nanoparticles, as it's called nanoparticle LD mass spectrometry enhanced analysis. And these nanoparticles are used to capture metabolites. And again, the result is then subjected to mass spectrometry. Then we get a data matrix, so with discovery cohort and the validation cohort, and based on the data coming with the patients, so we can then predict or determine a pattern of metabolites that are predictive for the diagnosis or for the course of the disease. We also, the bioinformatics can also be very useful in drug delivery studies. In this case, we have so we are dealing here with a lung tumor and utilizing the lung microbiota. And we have here as a nanoparticles with the Saccharomyces pneumonia membrane coating, which is then inhaled in the mouse. And based on the distribution of the gallium nanoparticles here in the cancer cells, it can, so the best way of the drug delivery can be determined. A very important aspect where bioinformatics comes into play, so in combination with nanoparticles, is the test of the biocompatibility of the nanoparticles, which is a huge topic. So in this case, the approach would be so that we have some nanoparticle treatment and apply it to either cells or a mouse model or whatever. And then the messenger RNA is isolated, and then as we see the differences in the mRNA expression between the treatment and the non-treated group, and based on the differences in this mRNA expression, as we can then deduct as which pathways are activated as by the nanoparticle treatment. In this case, this would be, for instance, as here, a typical gene enrichment analysis with, in this case, the P3K pathway activated protein description, absorption, focal adhesion, etc., so with a pretty high gene ratio. And this pattern of activation then will show us as whether the nanoparticle is biocompatible or not. And in any case, so how is the activation pattern of this nanoparticle in the particular cell model? Okay, so I think I will come to the conclusion now. And the conclusion is, so the take-home message for you is that we have a great synergistic potential. So with the integration of omics technologies with nanomedicine, and this might overcome quite a lot of limitations in current cancer therapies and other diseases, so by enabling precise patient-specific treatment strategies. Then we have an improved personalization because the omics data provide critical insights into the tumor biology or the biology of the disease, which will support the design of nanomedicines that are tailored to the genomic, proteomic, or metabolomic makeup of the individual patient. And there is also the, let's say, the view of an enhanced therapeutic outcomes with omics-guided nanomedicine that enhances both the efficacy and the safety of cancer treatment, because it enables tumor targeting more accurately. However, there are some challenges. So, as means, so that the clinical translation of omics-enhanced nanomedicines still faces challenges such as data complexity, standardization, and regulatory hurdles. However, I think so that the advancement of the bioinformatics technologies in combination with nanomaterial engineering will greatly, let's say, enhance these or greatly address these challenges. And by the way, this QR code here, so if you want to contact me, so then this QR code contains the contact information. Okay, so I think that's it.
Great. Thank you so much, Thomas, also for your very nice presentation. And actually, that was it already from NanoMedicine Austria side. So I'm happily handing over now to NanoMed Spain. First speaker will be Cristina Arimany, presenting the NanoMed Spain platform.
So thank you, Susanne. I will be brief, mainly for two reasons. First of all, because you will see that the work that we are doing from the Spanish platform is very similar from what they are doing from the Austrian platform. So Susanne already explained many of the actions that we are doing, but I will put some examples. And second, because you see that we had many researchers, many Spanish researchers who were interested in participating today in this session and sharing with you their research. So I will give space for them, who are the ones that are really doing the research in Spain. So the Spanish platform, as Josep said at the beginning, is turning 20 this year. So we have been running since 2005. And our mission is to accelerate the adoption of nanomedicine in the clinic and the market and bring together all the relevant stakeholders. Our vision is to bring together public and private collaborations, also to support the participation in international projects. We also have an expert advisory group that collaborates with the European Commission to give them feedback about the working projects. And we also aim to disseminate our results of nanomedicine to the society. Right now, we have more than 190 members. Half of them are from investigation centers, and the other half are divided between companies, technological centers, hospitals, and also observers who are mainly funding agencies. In this map, you can see that we have members from all over Spain, and mainly from those poles more active in innovation that are Catalonia, Madrid, the Basque Country, and Andalusia as well. We have been working in preparation of some documents that you can download in our webpage as well. The last one that we prepared is this booklet where we show the participation of all the Spanish groups that got a project at EuroNanomed. And you can see there the research they did during this call. As I previously mentioned, we aim also to make a difference in the working programs from the European Commission and make sure that they include nanomedicine in these calls. So we participate in a consultative group, and we gave our feedback on the European Mission on Cancer, also in the Horizon Europe Cluster for Health, and also in the Innovative Health Initiative. And we are also very active collaborating with other technology platforms, just like the ones that we have here about pharmacy, some health technology, printing, veterinary, and also with the European Technology Platform. As I mentioned before as well, the Spanish researchers were very successful in this EuroNanomed call. And from the 61 projects that were financed, 35 of them had a Spanish participation. So this was a great success. And we know that the Spanish researchers also got very good results in the Nanotech MEC call, and we will be happy to share also them with the community as soon as they are published. We also want to make sure that the research that we are doing at Spain is known internationally. That's why we participate in events like this one. And we also participated last summer in a mission that CDT from Spain organized to Thailand with some companies and also some research groups to explain the research done in nanomedicine there. Our scientific coordinator, Josep Samitier, is also very active in the field, and she is member of the advisory board of this Preciseu project, a European project about personalized medicine. And he also participates in international forums, just like this one from the European Commission on Advanced Materials for Industry, and this other one organized by the ENL about Advanced Innovation Materials for Health. We organize many events during the year. I will just give you a very brief overview. We organize joint events with the other platforms. We are going to have one next May in Madrid with all the platforms who do research related to biomedicine, and also with the platform funded by the Instituto de Salud Carlos III that are more related to clinics, with our aim also to promote translation. And we organize also events on the World International Days, such as the Nano World Cancer Day and the Nano Rare Diseases Day. And we do this with the aim of approaching nanomedicine more to the society and letting them know all the potential that nanomedicine has in the treatment and diagnosis of illnesses. We'll also want to inform our members about funding opportunities, and that's why we all organize webinars, just the ones like we have here about this call on public-private collaboration, or we organized one last January about possibilities to collaborate with China and other calls or public tenders. And also webinars or seminars on site of topics that are of interest for the community, such as open science and intellectual property, or sustainable research. And also we participated in one about the involvement of nanomedicine in veterinary. With the aim to make nanomedicine known for the society, we developed some years ago these education materials for schools that teachers can download and use them in the classroom. We have them translated in English, so if anyone is interested in downloading them, they can download them for free in this webpage. And we use them to organize writing contest where students from 9 to 11 years old could write and draw their comics or stories about nano drugs. We want to keep our members informed. That's why we have a monthly newsletter in which we inform them about the latest research done on the field in Spain and also about funding opportunities and the next events. If you want to get this newsletter, you just have to register and you will get it. And also we have a web page where we share all the information, and we have social media. We have X and LinkedIn. As Josep already mentioned, and also Susanne said that they will be participating, we are now organizing this big conference that's going to be held here in Barcelona next May, and we will be happy to welcome you here and talk to you there. The coordination team is composed by Josep Samitier, who you already met, Teresa Sanchez, who is very sorry that she couldn't come today. She was the one who had to give the presentation, but she couldn't make it at the end, and myself as project manager. And now we also have people from communication helping us with the event and the web page. Thank you very much. It didn't change. Okay, so here is our email if you want to contact us, and if you follow this QR, you will get to our webpage where you can register to get our newsletter. And if you are not member yet and you would like to be member, you can also become member following this link. Thank you.
Thank you very much, Cristina, for this very nice presentation. Always impressive what you have already achieved in NanoMed Spain in the last years. And now we hand over to the scientific presentations from NanoMed Spain. We start with María Jesus Vicent, sorry for that, talking about polypeptide-based nanomedicines, enhancing tropism and overcoming biological barriers. Perfect.
Okay. Thank you very much, Susanne and Cristina, for the opportunity to participate today. And what I prepare is just a few of the projects that we are running in the lab as a selection, just to see to foster these collaborations and capabilities. So we are based in Valencia in the Centro de Investigación de Felipe. And this is the most important slide, where we have an interdisciplinary team working on trying to identify new therapeutics or biomarkers for unmet clinical needs. And our strategy is trying first to understand the disease. So we collaborate a lot with clinicians and not only for understanding the needs of the patient, but also for getting the samples to really make these preclinical models that really resemble the aspects of the disease that we want to target. But we have also many chemists, and indeed myself is also a chemist in the lab and material scientist, where we have our chemical tools that I will show you some examples where we are mainly working on polypeptide-based materials, where we, after understanding the disease to target, where the molecular target is, and also the biological barriers that we want to really bypass, we can really engineer different type of polymer therapeutics, polypeptide-based therapeutics to reach the goal. And in the process, we are also looking for functional biomarkers of nanomedicine and trying to really facilitate or really to speed up the process. I don't know if you see well the screen. Could you see the screen, full screen?
Yes, everything perfect. We see it well.
Good. So why we are working on polypeptide-based materials? Polypeptide-based materials is we know how to control the chemistry. We know many years ago we established in the lab the NCA ring opening polymerization approach, where we established a spin-off company that they are now specialized, or is a CDMO, is Curopath, is more than 100 professionals working there now, because these materials are biodegradable, biocompatible, multifunctionals, and the chemistry, as I mentioned, we can control because we implement a new way to start the polymerization to control the starting and the end functionality, and also the solution, the conformation in solution, the topicity. And we are tackling many diseases. Some of them are metastasis in cancer, for example, but others are chronic diseases like neurodegenerative disorders. So it's very important if these materials are biocompatible and biodegradable, because in the way to have the metabolites that are endogenous is very important. So having this biodegradability is very important, and also the polypeptides offer a huge variety of amino acids we can use, and these amino acids also can go from something that is completely innocuous in the organism, so we can have systems that are hydrophilic and with stealth properties, but can go also to some amino acids where we can trigger some specific responses. And indeed, we have some projects where these materials are able to modulate and to synergize with the active principles that we are conjugating to the carrier. So also these materials are able to have different properties, conformational, as I mentioned. So we not only synthesize, but also depending on the properties, we can arrange and can trigger some different supramolecular assemblies that we have materials to go from 8 nanometers to 100, more than 100. And also we are implementing chemistry that allow us to have a specific control in this biodegradability, not only the material itself, but also in this self-assemble and the stabilization of the supramolecular assemblies. This control on the cross-linking and also some of the conjugation that we are doing in the materials, we are now implementing in process control like microfluidics, and that allow us to really increase the possibilities to biospheric reproducibility and fostering the control towards the patient. These materials not only are starting as a whole biomaterials, like as amino acids, but also we have some projects within the complementary scheme of the materials line, where we are starting with some macrocycles with inherent properties, inherent imaging properties, sorry. So we are also working towards the diagnostics, and this diagnostics has dual applications for us. So we control the chemistry, as I mentioned, with the polymerization, but also as the macro initiator is a porphyrin or phthalocyanin, we can monitor where these materials are going, but also we can implement combination therapies because we can complex some metals in there, so we can use for not only pharmacological activity, but also photodynamic therapy or some MRI or other type of radiosensitizers. So because there are tumors that are recurrent or resistant to even radiotherapy. So we are involved in a line of projects where the construction of these materials to transport small molecules, but also gene delivery now, most of them. We are looking at prostate cancer, that is like an advanced prostate cancer, breast, ovarian, but also with these materials, if we decorate also the surface and we use different type of vehicles, we can reach also the brain through an intranasal administration. And we have already proof of concept of several of these. And for example, for the intranasal, as you see here, if we embedded our nanosystems in a hydrogel that is based on hyaluronic acid cross-link through polypeptides, we are able to really bypass the BBB, because the intranasal administration, we don't have the BBB itself, to have a nice biodistribution in the brains with more sustained release and accumulation in different areas of the brain. So we are exploring this, not only glioblastoma, brain metastasis, but also we have proof of concept with some collaboration with the Vall d'Hebron in Barcelona and the University of Barcelona. Also, we have proof of concept in Parkinson and transporting biologics like the GBA protein, for example. Other barriers that these nanosystems can really cross, and also playing with the size and the shape of these materials, is if we have something that is around 100 nanometers with highly negatively charged, they are really like the immune system. So we discovered that some of our materials are able to accumulate in the lymph node. They are renally excreted if they are negatively charged, but also it really has this size around 100 nanometers and minus 60 in the zeta potential. We accumulate in the lymph nodes, and it's open up possibilities of cancer nanovaccines. And we have several projects in La Caixa in pancreas, where we have this nanovaccines accumulation in the lymph node, triggering the production of CD8 T cells, and also the possibility to modulate the tumor microenvironment, and which has recently published this year the first proof of concept in melanoma, also in collaboration with University of Lisbon and also with ENEO and Xenopath, where we are able to we are conjugating a multivalent vaccine, so the two different antigens, for example, in the surface, TP100, MHC1 and MHC2. We were able to have this tropism in the lymph node. So we accumulate this a cut on the lymph node, where we nicely accumulate and distribute in the lymph node, and also target not only the dendritic cells that you just go to the paper and have the whole mechanisms, but also some of the endothelial lymphatic vessels and also blood vessels that really allow us to have a very nice activity, even in a xenograph, in a patient-derived xenograph model, with a nice immune response. So this is just playing with the size and the shape and the zeta potential of these materials. We have different tropisms. But we also recently published a paper where several papers where not only we are reaching different organs, but also we can go to intracellular organelles. So this was a project of a training network, Marie Curie, just finished, and we have continued now with another one called NAT-Prime, that we are continuing with this work to achieve proof of concept in vivo. We have a diblock system that has an inner tropism to the mitochondria, because they has a high affinity for the cardiolipin. This acts as an oligomer that independently where it's conjugating, it really drives things into the cytosol. So we are exploiting this approach. So you see here a very nice example where this small oligomers, a diblock of polyproline polyornithine, that is scaled up now in a very large scale with Curapath, the company that we established many years ago now is a CDMO in Valencia. So it has a nice co-localization and a very nice, this is a collaboration also with the Master Institute, the Latvian Institute of, where we have really this, even in the physical model with cardiolipin, we have a very nice interactions only with the cardiolipin is present in these vesicles. The uptake is not only by endocytosis, we have a direct uptake that is going directly in the mitochondria with a very nice response at the mitochondrial level, and because these systems are able to interact with the complex one and modulate the OX4. So we are now moving forward with these oligomers in some of our conjugates to really target and to really identify target in the mitochondria and do some transfection to have this key performance indication that we need to move forward with this material towards the patient. In all the process, we characterize very deeply, and we implemented techniques like the AF4 or LCMS, and also now we are working with MS imaging, and we are very keen on understanding the response in all the parts of the design, not only the materials, but also how we link the drugs. So the linking chemistry and the release kinetics is very important in all our design. So this is the whole process, but also characterizing very well the models. As I mentioned, we are interested into microenvironment in the regenerative disorders. So the environment and the models itself are very important. So we work, for example, with 3D heterospheroids and also patient-derived organoids. And now we are involved in European projects like Unlock or Impulse, where we are really looking at this organ on chip and to have something that resembles before going to the in vivo. When we go to in vivo, we also characterize well the models. We have, for example, models of breast cancer, metastatic breast cancer fully characterized, intratibial models of prostate cancer, or orthotopic models of prostate cancer, for example, among others. And we have techniques to look at specifically to microenvironments. So we are really happy to collaborate and to foster interactions either in the chemistry side with the application or offering other models to really test a type of nanosystems. And we have already many collaborators and funding agencies, and thank you so much for listening, and looking forward to it, to any collaboration. I have to leave, sorry, so if you have any questions for me, please put it in the chat and I will reply by email. Thank you so much.
Perfect. Thank you so much, Maria, for your presentation. Now we will continue with Rosario Sanchez, who will talk about nanomedicine network to advance the diagnosis and treatment of high-impact diseases.
Okay, so I will just take a few minutes, because what I wanted to do today is to take this opportunity to present the network NanoCare 2.0, the second generation, where we are working in nanomedicine network to advance the diagnosis of treatment of impact disease. So this network has been funded by the Spanish government, and what we are trying to do, as you can see, so what we have here is a network for an institution that they are focused in different subjects that I will go very quick share with you. But the main idea, as you can see here, is to collaborate to try to go further to the translation of the clinic. So the aim of the network to coordinate our effort. The main idea to be able to don't repeat the same mistake and to finish this, accelerate what is going. So we are working with different materials that I won't go through all this chemistry or this a platform and experiments, but just to have a view of what we are doing. We have several materials, and the thing is we have divided in 5 research lines. So we are some experts in neurodegenerative diseases, some of them expert in cancer diagnosis. Another part of the members of the network are experts in infectious diseases. And then we have 2 more research lines that we have wanted to go further and be focused in characterization of nanomaterial to reach clinic. And the last one, you go from the bench to the market, like to complement these 2. One will be like transversal to the other ones, all of us. So here, very briefly, just to show that there are teams for many important research institutes around Spain related to medicine. I won't go through that. You want to go contact me. We have the workflow, but the kind of activities that we are organizing through this year. We are doing some face-to-face meetings, and we are building some seed cooperative project. We have small funding calls inside the network to help to build these collaborations, and then have public awareness. We are participating in many activities, social and scientific missions for the junior nurses of the network, and then all one transfer So I won't go through that just for you to think construction, different standard to work. Just for sure, always counting with the support and the help, the European Technological Platform, TPN, and for sure with NanoMed Spain. That's great. And just if a couple of things just to know, one of the lines that we could collaboration with the Austrian network past that we have quite a lot of expertise and looking to collaborate, helping to characterize nanomaterials. So there are experts in different models. They brings the that that also counting with models more simple to work with, such as a elegant one. But the main idea is to try always to go through this portfolio of characterization, the clinical characterization further, with all the protocols standardized by the FDA, NIH, and the nanotechnology characterization laboratories. So we have building like a nanocharacterization lab. We are trying to help other people and materials. Also, we have the RL5, this research line. We are trying to help. We are organizing now a training school that will be happening in September. But the main idea is to try to prove expertise that we have already of what happened to us. We are some member of the transfer to help the other members. About this, we have page for I think that things have any questions. Thank you for your.
Great. Thank you very much, Rosario, for presenting NanoCare. With this, I would like to already hand over to, because we're a little bit behind the schedule, hand over to the next speaker, Samuel Sanchez, who will talk about nanomotors, self-propelled nanoparticles overcoming biological barriers.
Yeah, thanks for the opportunity and thanks for organizing this network workshop. So I will be talking today about what we do in our lab. Is about making nanoparticles that are moving around, that can be driven in solutions and overcoming biological barriers. And the reason it's very clear, and we all know here in this community, that there have been a big challenge for nanoparticles and nanomedicines to reach to the target, and it has been presented already in a couple of talks before. So there are many barriers, like at organ level, protein corona, extracellular matrices, and even like endosomal escape. So we try to tackle some of these, and in particular today I will be only talking about these matrix that we see around the cells and the mucus layers and everything that is surrounding, which is a big barrier that nanoparticles don't tend to cross. In that particular direction, we just published a couple of weeks ago a review on the use of macro nanomotors, which are self-propelled particles that can reach to the target, crossing hyaluronic acid, mucus, and cell membranes. And that has been highlighted in the cover. So what are self-propelled nanoparticles or nanobots? Are a form of two components versus nanoparticles that are things that we have seen, and everyone has a wide variety of experience in this community. We do particles from silica or polymeric particles, PLGA, hydrogels, nanogels, hyaluronic acid. So we have a huge expertise, and this is what you are seeing here. But the second component to make them move is an engine. So what we do is we decorate the nanoparticles around with enzymes, which take up the substrate, which is in the solution, and generate the chemical gradient that moves the particle forward, as you can see here. So passive particles, traditional particles that we normally do are not moving. They only have Brownian motion, but if these particles have urease enzyme, for instance, this is one example. We have many, and this is under is a patent that is now in territories like the decoration with different enzymes to generate motion, right? So if you have urease around and urea in the solution, this chemical combination will generate a fluid flow that pushes the particle forward. Okay, that is a technology we have developed over the last 10, 12 years. But one nanobot or nanoparticle does nothing by itself or little. You need billions of them. And this is what we are working through my ERC Consolidator grant, is to study the collective motion of millions of particles, as you can see here, as a smart drug delivery system. So one clear example is the case of bladder cancer. So if you have urea in the solution, urea is in the urine is in the bladder, and bladder is the most common cancer, the most expensive cancer to treat, because the treatments and the diagnostic are not efficient enough. So what you see here on the cover of Nature Nanotech is when we intravesically administer the nanobots, they move collectively, as you see on the left side, and they specifically penetrate and accumulate in the tumor tissue. As you see here, the yellow dots are the penetrated nanobots and not in the healthy urothelium. So this is the first case I wanted to tell you today, how they can penetrate. Just very, very briefly. So here's the final conclusion of the paper, where we studied by different techniques, MRI, PET-CT, confocal, and in this case, you see light sheet microscopy in combination with IRB Barcelona, where we have administered these motile particles, so not only passive, and these yellow-red dots are the nanobots, which manage to penetrate, as you can see here, by the scatter signal. So if we radiolabel with iodine, for instance, as a model API, we managed to see a normalized tumor reduction, a tumor volume reduction down to 90% if we have high doses, which still are on the limit on clinical trials. Low doses allows the stopping of the tumor progression. If you don't have any API, the tumor will grow. And if you don't treat, of course, tumor keeps growing after 1 week. So this is very unique, because normally patients go from 6 to 12, 14 times to the clinic. In this particular case, we reduce with 1 dose down to 90%. So with this technology in hand, we look at different biological barriers. And second is that we can not only do in vivo, ex vivo, or in vitro, we can do on a chip different models of extracellular matrix. We can have bladder on a chip or any type of organ on a chip, and tune the stiffness of the matrix and see the diffusion of these nanobots through and penetrating through this tissue in a very controlled manner. We also have, in collaboration with collaboration with ICMS in Eindhoven, with Vallebron, we had a project, and it's an ERC proof of concept as well, where we had, instead of urease, catalase. Catalase takes the ROS or the peroxide, which is endogenous generated, or you can co-administer, and we can cross the mucus layers. Here, here in red, you see the mucus, and on the left column is the passive nanoparticles, on the right column are the nanobots. So if the passive nanoparticle will get stuck, and they get very sticky to the mucus layer, they don't penetrate, but this catalase generates oxygen bubbles. They can propel and push and make gaps and voids and let the nanoparticles with the medicine crossing the mucus. And we are doing these experiments for the pseudomyxoma peritoneus, a very rare disease, and we are having good results in vivo. Then other places where viscosity is very high and therefore is a biological barrier are in the joints. So together with the Fundación García Cugat, who is an expert worldwide in the knee diseases for sports patients, we can see by photoacoustic imaging that if you administer in the joint, this is the joint of a mice, the nanoparticles, here is with the gold functionalized, they only accumulate at 1 point. But then if you have the nanobots, they have been spreading all around the joint, and this is better for the recuperation and the biophysics of biomechanics of the joint. What else? Another place where there's a viscous layer is the eye, right? Crossing the ILM in the ligand membrane is very, very challenging. So passive nanoparticle will get here on red, will get on the outer side. They never reach the cells. But nanobots are able, in this case, with collagenase and urease, they're able to degrade sequentially and very mildly like collagen, and then make these gaps that allow them to reach the cells. And the last case that I want to show today is how the skin can be crossed by nanoparticles, our nanobots here. Again, we have now in red the cells, in green the nanoparticles, and they don't cross and they start crossing. These are preliminary results, but they can start crossing the skin disease. So these are all the examples that we are, our group is particularly interested in. And here the message I would like to show you is that moving from passive particles, like traditional particles, to active nanosystems that can not only move, but degrade the extracellular matrix and the mucus is a very good tool for crossing biological barriers and improve the low efficacy that is nowadays in nanomedicine. Thank you very much.
Thank you so much for this very interesting, nice presentation. I would like to hand over to the next speaker, Montserrat Cano. Perfect. Montserrat, the floor is yours.
Okay, thank you very much. Yeah, thank you very much for inviting us today, because it has been really interesting so far. So I would like to introduce, I'm Montserrat Cano, CEO of NanoMedicine. I would like to introduce you to NanoMedicine. It's a biotech that we are developing targeted nanomedicines for oncology. Cancer, as you all know, is a very complex multifactorial disease with more than around 20 new cases diagnosed every year, 10 million deaths, most of them due to metastases and disease recurrence. So right now the therapy goes over the classic radiotherapy, surgery, and chemotherapy, and the most recent approaches to the disease is via multimodal treatment strategies. So incorporating breakthrough therapies like immunotherapy and targeted therapies that they are focusing on the cancer-driven cells. So if we look at some results of this targeted therapy, of the overall population that can benefit from it, it's still limited depending on the cancer type and the presence of the specific biomarker. And here I'm showing you the three main tumor types, like breast, lung, and colorectal cancer. And among these three tumor types, we can find only one biomarker common to the three of them, with a limited population that they have it. Also, even blockbuster antibody-drug conjugates that they are designed to deliver cytotoxic agents directly to cancer cells, they have important safety concerns still, mainly due to non-specific binding, premature drug release, or low percentage of drug reaching the tumor site. So we are developing a solution that is based on a unique proprietary platform technology that kills selectively cancer cells by a very unique selective distribution into the tumor that delivers a high concentration of drug into the cell, and it has a high avidity for those cells that express the targeted receptor thanks to the nanoparticle formation. And with all this, the aim is to get a safer profile for the drug, and we don't see macroscopic damage to healthy tissues in our studies. So our first candidate is targeting the CXCR4 positive tumor types. The CXCR4 receptor is well known in cancer. It's involved in tumor migration and resistance, and is highly correlated with aggressiveness and poor prognosis for the patients. And it's expressed in 40–50% of patients of more than 20 different tumor types. So if we translate these numbers into the examples that I showed you before, we can see that in breast, lung, and colorectal cancer, the subpopulation of patients that could benefit from our drug would increase importantly, and it can go even up to 10 million if we consider the rest of tumors that they also express the receptor. So our candidate is this NNL-1524 is based. It's a drug protein conjugate which has a CXCR4 receptor ligand exposed that self-assemble, forming nanoparticles. And the way that it works is that it recognizes the cancer cell and binds to the receptor on the membrane. It's internalized and kills the cell selectively. We have a full preclinical evidence for this drug, and first of all, mainly based on a GFP analog, we can show that there is a high selective distribution into the tumor, with more than 80% of the administered dose reaching the tumor. And when we administer this GFP analog bound to a cytotoxic drug, we can see that there is no macroscopic damage into healthy tissues. And Nanolegion, what has done is to take this original idea coming from university and develop it into a humanized scaffold and optimized drug protein conjugate, and we have already a full package of efficacy in both solid and liquid tumors, as I show you here in the slide 2 examples of them. As you can see, we are showing a high efficacy for all the models that we are working with, that all of them are CXCR4 positive. And our aim is to progress this drug into clinical trials, and this is mainly the Series A that we are fundraising and aim to close soon. And I wanted to finish this presentation by introducing you the team. So and the founders of the originators of the technology, coming from University Autónoma of Barcelona and Institute and Sant Pau Institute, are founders of the company, together with our chairman Manuel Rodriguez, who was the entrepreneur. And together with me, we have more than 20 years of pharmaceutical drug development. And also, lastly, financially, we've been supported by the 3 investors of our seed round and key opinion leaders in drug development in oncology. So thank you very much.
Great, thank you very much. Very interesting. Thanks again. Now I would like to hand over to Rafael Gomez Ramirez and his presentation. Excellent, all fine now.
Yeah, okay. So I was saying that thank you so much for this really nice webinar. Well, I would like to show you what we are doing in our group in the University of Alcalá, which is very close to Madrid. And we are a group that we are preparing, synthesizing the dendritic system for different biomedical applications. And we are working mainly in carboxylene dendrimers. Why carboxylene dendrimers? Mainly because of 2 reasons. The first one is because we have, let me put the laser. We have a very lipophilic skeleton, and we have a lot of data that show us that we can cross physiological membrane more easily than hydrophilic systems. Physiological membranes are also lipophilic. But to give water solubility to all these systems, because they are very lipophilic, we need to include charged groups or very polar groups in order to give water solubility, but also the functionality of these groups show us the different biomedical application. And we are working in cancer, in antibacterial, in a lot of diseases. Another property with this carboxylene dendrimers is because we are using low generation dendrimers. You can see here that we are using a small molecule, generation 1, generation 2, generation 3, where we increase the size, and also we increase the number of functional groups that we include at the periphery. This is what we call multivalency. In general, the use of low generation means lower toxicity. If you compare this with, for instance, permanent or big polymers, in general, the size increase the toxicity. So working with low generation reduce notably the toxicity. Another important fact is the dendronization process. If you see this spherical dendrimer, where we can use this spherical for as an anti-cancer or as antibacterial, depending on the functional group that we have here, if we take a piece of this spherical dendrimer, what we end up is with what we call a dendron, where we have the same kind of functional groups, and even the same number depending on the generation. But what we have here is a focal point where we can use it to bind to different nano-sized platform. In this way, we have working with dendronized nanoparticles, dendronized polymer, for instance, and also we have created a dendritic method, because if we put, for instance, here a long lipophilic chain, we end up with dendritic method. We are really robust comparing with normal method. At the same time, we can prepare, if we link two dendrons, we can end up with Janus dendromer, where in one side or in one phase, we can include, for instance, therapeutic groups, and in the other phase, we can include targeting groups. But what we are interested in this moment is to study the use of metallodendrimers. I mean, to include out the periphery different kinds of metal with different kinds of activity, mainly as antibacterial or as anti-cancer. What we used to do is just to study the monometallic, and then when we know that they have a very good therapeutic behavior, we transfer this information to the dendritic skeleton in order to work with the multivalency. I would like to show you two fast examples of what we are doing. When we're using silver metallodendrimer, the mode of action or mechanism of action is completely different than that comparing with cationic system, dendrimers or polymer. When we use cationic dendrimers or polymer, what we have as a mechanism of action is a very rude way to destroy the bacteria through the polarization and pore formation. But when we use silver metallodendrimer, the mechanism of action is smoother, just delocalizing proteins or fatty acids which are in the bacterial membranes. So we can see a different behavior. Another example is, for instance, when we include here ruthenium complexes. When we use these ruthenium complexes, we are using this as an anti-cancer system because of their antioxidant properties. And we have studied these systems in the interaction with the hypoxia-induced factor, which is a factor really important for cancer cell, in the survival of the cancer cell, because, for instance, they create a high vascularization for the survival. When we use our metallodendrimer, this activity decreased, notably decreasing also the vascularization and therefore decreasing the survival of the cancer cell. But also we can observe using these systems other kinds of antiproliferative properties, like for instance, reduction of the expression of cancer stem cell, which is really, really important. What we are doing at this moment is to use this behavior of the metallodendrimers, mainly the redox activity, in order to work as antibacterial or as anti-cancer strategy, a new strategy called catalytic biomedicine. What we want is to use the multivalency in order to create an imbalance in the redox homeostasis of the bacteria or the cancer cell, just provoking an oxidative stress or a reductive stress, depending on the condition that we are working, and then affording a dead cell of the bacteria cells. So with this in mind, which is our focus in this moment, we offer a synthetic platform of dendritic and metallodendrimers to access to innovative and multifunctional material and new modes of actions in different kinds of of biomedical application, mainly as antibacterial and anti-cancer, because we are focused on this. And what we are looking for our Austrian colleagues is to find experts in biological, biomedical, or pharmacological issues that can feel comfortable in the use of our metallodendrimers for the search of new mode of action within this catalytic biomedicine approach. So I think it is all for now. Thank you so much for your attention.
Great, thank you very much for this nice presentation. With this, I would like to hand over to our last presentation for today, to Oscar Castaño.
Thank you very much for your invitation. My name is Oscar Castaño. I'm senior researcher at Institute of Bioengineering of Catalunya, IBEC, and also I'm tenure associate professor at the University of Barcelona. I work. I collaborate in very close to with the group of biomaterials for regenerative therapies led by Professor Elisabeth Engel. And I must say that we are not working in nanotechnology, but we use nanotechnology as a tool in practically all the lines we are working on. So this is my presentation. It's titled Nano Tools for 3D Microenvironment Engineering. So we go from regenerative therapy innovation to nanotechnology evaluation of models, and I will explain why. So this is our mission, is basically development of knowledge and technology. So we are an applied science group, although along our along our journey, we find a lot of fundamental science holes that we need to fill. So we develop the capability to do that. So we work in both fundamental and applied science with the goal of transfer the innovation of biomaterials and scaffolds and to create these microenvironments for tissue regeneration. So this is what we are focused on, are bioactive biomaterials and the creation of nano signaling provided platforms through basically nanoparticles. Also the introduction of nanotopographic signaling, modifying surfaces, especially using electrospinning, and the methods we are working with. We are experts like sol gel, water oil emulsion, nanoprecipitation, as I mentioned, electrospinning, and also nowadays we are specialized in the submicronic 3D printing by two-photon polymerization. So the goal is, as I mentioned, creating microenvironments for tissue engineering and translate them, this basic research, into new development products. And then we saw that just to test our own microenvironments, our own biomaterials, there was a huge gap between in vitro analysis methods and the in vivos. So we started in 2016 to develop our own in vitro models that then we also offer for drug screening, both in nano or the scales test. So electrospinning, we have many articles talking about that, using electrospinning as a very cost-efficient way to create nanostructured extracellular matrices, like this one, where by PLA, amorphous PLA polymeric nanoparticles, we were able to vascularize the brain of neonatal mice. As well, we are able to integrate nanoparticles, signaling nanoparticles, ceramic, both ceramic and polymeric, into nanofibers, just to create platforms both 2D and 3D in polymers and hydrogels. We are working extensively in models and scaffolds that are in between the 2D and the 3D or the 2.5D using electrospinning or extrusion bioprinting. And nowadays we are focusing on photo-based 3D printing like DLP, stereolithography, or as I mentioned before, the two-photon polymerization. In terms of research line, we are working with extracellular matrix-like scaffolds for skin regeneration. This is we have a patent nowadays which is granted and licensed to a company, SL, to the treatment of diabetic ulcers, because one of these fundamental holes we had to fill is how calcium concentrations between a very tiny window range of concentrations were able to stimulate the angiogenesis of different mesenchymal stem cells that then, by crosstalking, generate angiogenic sprouting in blood vessels. As well, we are working on the creation of nanoparticles, both ceramic and radioprotective nanoparticles of PLGA containing amine phosphine for the treatment of oral mucosa defects after a tumor resection, and especially targeting the protection against ROS species after these treatments of radiation. As well, we are working in the scaffolds for bone regeneration, both in terms of nano or submicronic extracellular matrix and 3D macroscale scaffolds. As well, signaling for molecules for cardiac regeneration, especially lactate signaling, in terms to try to regenerate the especially tissues after infarct. And then we moved 10 years ago into organ-on-a-chip models, as I mentioned, to try to assess our own materials, but then we opened for other, because they were very flexible. For example, we have these models of angiogenesis on a chip, just to assess proangiogenic biomaterials, but also now we are working in tumor models, because in the end, tumor is probably the most proangiogenic, let's say, tissue or material that we know. So it's a very good proof of concept for us. And then myocardium on a chip, starting to try to model infarcts. We finish as well working on the modeling of tachycardias. So now we are trying to go on 3D in these tiny models and in microfluidic devices. We are also controlling the mechanobiology signaling by working on the ink by 3D printing and two-photon polymerization, also using simulation by finite elements to try to expect or close a very narrow range of working materials. We work as well in vitro 3D cancer models for drug screening. And these 3D cancer models we try to personalize using decellularized extracellular matrix, both from decellularization of tissue or creating our extracellular matrix by stimulation of cell-derived extracellular matrices. And then this is useful just to test for drug screening against tumors, but as well it's open for nano drug or nano biosystems screening. So this is our group. She's Elisabeth Engel. We are a very highly interdisciplinary group with a strong team working. We collaborate both with clinicians and companies, and we are funded by both national and international projects. So here you have a QR code if you want to check our website. Happy to collaborate with all of you and try to find ways to just interact. Thank you very much.
Thank you so much, Oscar, for your presentation. So with this, we close the part of the scientific presentations also from NanoMed Spain. I would like to thank all speakers, both from Nanomedicine Austria, but also from NanoMed Spain, for their great presentations. It was super interesting. It was the first joint webinar between Nanomedicine Austria and Spain, NanoMed Spain, and we decided to keep it topic-wise rather open. We will think about if we continue with this effort and if we do a second edition at some point. If you have any ideas or suggestions of specific topics that you would like to have addressed, please reach out to either Cristina or myself or our colleagues so that we can also follow up appropriately.
Thank you to you for all the organization and the logistics. And as you said, we will be happy to continue collaborating and to organize maybe thematic webinars, because today it has been very broad, just to give an overview of all the possibilities that we have. But it may be an option to organize thematic webinars and also on topics of interest for both communities, just as regulation or green chemistry or any topics that can be of interest.
Yeah. Excellent. So with this, I would like to thank again all speakers and participants for your time and your efforts. It was a very nice experience also for myself. Thank you very much. And yeah, I wish you a very nice weekend already.
Thank you, Susanne.
Thanks
Thank you
everyone
very
for
much.
coming.
so much for the
Bye.
opportunity.
Bye.
Bye.